Hello, everyone, and welcome to today's broadcast, a keynote presentation, Solid Phase Microextraction, New Developments in Bioanalysis and Medical Applications. It is presented by Barbara Bojko, PhD, who is an Associate Professor at Nicolaus Copernicus University in Torun, Poland, and Janusz Pawliszyn, PhD, who is a Professor of Chemistry at the University of Waterloo in Ontario, Canada. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before I begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speakers will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Bojko and Dr. Pawliszyn. I will now turn the presentation over to them.
Thank you very much for the kind introduction. So I will be talking first. My name is Janusz Pawliszyn from the University of Waterloo, as I was introduced. And in my part of my presentation, I'll focus on fundamentals, just to give you an idea why the technology works and what the fundamentals of the technology. So when you look at this presentation, you need to have open mind, because we'll be changing paradigm of present way of doing analytical chemistry, specifically sample preparation. So I will try to explain you something which might be looking difficult to you. In this case, solid phase microextraction, which you might not practice on every day, and it looks therefore it's difficult. But I will try to make it as simple as possible. I'll point out the opportunity to improve the understanding of the technology by further courses or papers. So again, the idea is to look out of the box. What you do presently is not necessarily the most optimal. The science moves forward, and therefore you need to consider your options. So the technology I will focus on is in the area of sample preparation. As I understand, most of you are practitioners of analytical chemistry, so you understand that this is the process. The analytical process consists of several steps. So I will focus on the area of sample preparation. So ideal sample preparation would facilitate high throughput or in vivo sampling. That's what the Spme does. It requires a very effective coupling between the sample preparation, and convenient introduction to the separation device, such as chromatograph or directly mass spectrometer. Traditionally, and many of you probably consider sample preparation as an art, not a science. However, the progress in science allows us to look much more carefully on different processes occurring when you do sample preparation, and therefore, you can actually have a full understanding of what you are doing. So you can rationally optimize the process to be very effective. And by doing so, of course, you have a lot of advantages. I understand many of you are working in clinical labs or industry, don't have the time to spend on it. But of course, I work in academia. I had the opportunity to work with a young scientist for a period of their thesis, where we can make a scientific project and therefore have the depth of understanding. And by doing so, we can make it as interesting as possible. So we can look at the subjects in vivo, as you will see. We can look at the very difficult samples, get the very interesting information about the system. So basically, the choice of your sample preparation will allow you to drive the amount of information you're going to get. And of course, many of you, as I am, are concerned about the environment. So anytime you consider a new area of sample preparation, new technology, you need to consider the technology which are green in nature. This means eliminate organic solvents, eliminate reagents, do measurement on site and in vivo to facilitate measurements directly where the sample is. So you don't need to transport the sample to the laboratory, which of course is energy consuming step. So it's not only elimination of solvent, but also moving the analytical chemistry on site, basically performing the screening and on-site analysis. In some cases, like medical laboratories, is very important because it gives you immediate information to the doctor or surgeons about the condition of the patient. Now, of course, I'll be talking about extraction. So I will just remind you about the very fundamental process of partitioning. Yes, so if you have a, for example, sample made in contact with the extraction phase, analyze partition into the extraction phase. If you choose extraction phase, you have higher affinity towards the analyte than the sample matrix. So you basically have this enrichment factor K, which is listed below, which is basically ratio of concentrations at the low concentration. So you basically have a process of increasing the concentration or enrichment. So, but what I'll be focusing on, it's not a traditional exhaustive extraction technique, such as liquid extraction or protein precipitation, but I will try to encourage you to consider micro extraction. So this will be very important for you to understand the difference and the advantages. So this is the example to clearly demonstrate this. So here we have water analysis. Yeah? So typically in water analysis, you have 1 liter of water, 40 milliliters of solvents, and basically then you separate this and do further evaporation, so have a concentration, then you concentrate to 1 milliliter, and then you inject 1 microliter out of this into the analytical instrument. So it's a very, very lengthy process, uses complex organic solvents, and of course evaporations and what happened with the vapors. Of course, it goes to the atmosphere, which eventually we breathe. So what I'm saying is, let's replace it with a much simpler process. You have for example, fiber coated with about 1 microliter of the extraction phase, some type of solvent, and place it in the same amount of 1 liter of water. Now, obviously, some of you says, well, you can do it, but what about the sensitivity of the measurements? And in fact, if you go through the calculation, you find that the sensitivity will be identical. And why? The reason why is by trying to exhaustively extract using 40 milliliters of solvent, you actually lower the concentration in water. And therefore, as you remember from the previous slide when I show you distribution constant, the concentration in water is less. So therefore, the concentration in the solvent is less as well. While here, if you use a small amount of extraction phase, the concentration in water doesn't change. So your concentration of the fiber is much higher. So you have much higher enrichment factor. So this is one advantage. So you have high enrichment factor. And the other advantage is that you don't need to define the volume, because if you do exhaustive extraction, you need to know the volume to know that you will exhaustively extract. Well, here, you basically place it in the source. We'll see, we talk about tissue, we talk about bloodstream, we talk about another application, environmental application of the water, and so on. So it's basically, you don't need to measure the volume of your sample, which is very convenient for on-site analysis. So this is the major reasons why microextraction gaining interest. So I will particularly focus on one form of microextraction called solid phase microextraction. And this is the technology where the extraction phase is the sorbent, which is dispersed on some type of support. And the amount of the sorbent is low. So, as I discussed before, you don't do exhaustive extraction, but you rather extract and calibrate the equilibria or some kinetics factors, like it would be, like I mentioned below, in flow injection analysis. The beauty about SPME is not only what I say has the advantage of enrichment, but it's also you can coat any type of surface. Yeah? So you have a surface of the fibers, you can see surface of the tube, surface of the vessels, particles, steering mechanism, disc, and all of them has been already investigated. You see the publication below, which kind of introduces to those different topics quite early. And in the previous slides, you see the references to the books which has been published on the topic. So if somebody is interested, please consider getting those books. Like in this here below, you have another book of Theory and Practice. So if you use the fiber format, which is the most popular one, but it's not the only one which we use, but most of the application will show you here will be fiber, but also the blades. I will show you in a moment. So basically, you can consider 3 different types of experiments. Direct extraction. So if you have on the first slide from the left, so you have a direct extraction pre-matrix, headspace extraction, which is very powerful application of the SPME. Or more recently, and that's what I will focus on, looking at the very complex samples and using some type of membrane protection approach to avoid the contamination of the coating by real matrices. No, the SPME is commercially available, primarily for you looking at the food analysis in fragrance type of compounds, but also in clinical also applications, residue, for instance, solid residues and drugs, and it has been used in headspace of blood and so on. So it's a very complex matrix that can be analyzed that way because you isolate the sample from the extraction phase via headspace. So the device which is available is called Velco SPME device, and is shown here. It's basically a syringe where you have a coated fiber inside the needle. By moving the fiber in and out, you can expose the fiber to the sample, or you can protect it in the needle. So if you do extraction, you expose the fiber, or if you do thermal desorption, you expose the fiber, and if you penetrate the septa, you keep the fiber in the needle. And typical automated device is CTC autosampler, which is sold all over the world. This is another opportunity for you to learn more. There was a recent paper, Analytical Chemistry, published in January this year, describing the recent developments in the technology. As you can see in the center, you see the syringe, and there are different approaches, including the drone. You can see underwater sampling, doctor's office, in vivo sampling. We actually have the human trials now. So it's basically a lot of different opportunities because this technology facilitates that. So I will tell you now about looking at the complex samples because that's what we focus on our presentation. So as I mentioned to you, the very popular application of solid phase microextraction is the headspace analysis. As you can see, in the headspace analysis, you separate the extraction phase from the complex samples via headspace. So anything which is volatile goes to the fiber coating and is enriched there, due to the K value. Remember what I discussed about. Then you can take it out and desorb in local instruments, and you get a good clean extract, because anything which goes through the headspace is clean to be thermally desorbed in the auto-injector of the GC and then separate in the column and detect it. So we have been working for the last 20 years to develop this direct extraction. So you can see below, we have fiber placed directly in the matrix, and we wanted to also achieve this cleanup approach. So how did we do that? So you can see the matrix and the compounds bound to the matrix. They are in the equilibrium with the free concentration of the same analytes. And now we are trying to equilibrate the coating with this free form. So in fact, anything which goes via free form to the coating, as long as those matrix the matrix shown here like proteins or pieces of tissue do not adsorb on the fiber, you have very clean extraction as well, because it goes via free form. This means it is reasonably soluble in water, so we can dissolve it in the LC and LC injector or the solvent and then introduce it to the LC and basically perform the separations on the clean matrix. So here, the example, what will be benefit of that? So you have a apple homogenate. If you look on the left slide, this is headspace analysis, 60 minutes extraction, and the comprehensive two-dimensional gas chromatography. And you can see that compared to the right trace, where you have again two-dimensional chromatography, comprehensive two-dimensional gas chromatography, on vertical axis you have a boiling point, and on horizontal axis you have a polarity. So you actually see much more compounds, yes, on the right side, especially high molecular weight and polar ones, because those have low Henry constant and therefore they are not extracted very well by headspace approach. So how do we now do that? Incorporate this approach, direct extraction and clean extraction. So basically here it is shown what we do. So we basically have the fiber, as you can see up in the upper part of the slide, which has a coating. The coating is surrounded by the protection layer. Okay? And this protection layer, as is shown below, can consist of the Teflon, for example, polychloroneutral or PDMS. So if you have such a system, because of this protection layer, nothing is being adsorbed on the surface of this polymer. So you have a clean extraction. So you just need to have rapid rinse with clean water and perform the desorption. Now, in addition, this technology works very well from practical point of view, produces so-called balanced coverage of hydrophobic and hydrophilic compounds at the same time. This means we can extract compounds from different log P, as long as the sorbent has high enough polarity to go very deep in the polar range. So basically, why does it happen? So here the slide demonstrates that. You have this fiber with extraction phase. In addition, around the fiber exists so-called boundary layer. So it is the static layer. This exists in any type of system placed in a fluid or tissue. So in order for the high molecules to go through, it takes a long time. So within a half an hour or so, or less, when we perform the sampling or blood extraction, whatever, those do not typically reach the fiber. If they reach it, as I mentioned, the fiber surface is impregnated with the polymer, which prevents its adsorption. So by rinsing it, you can get rid of it. Meanwhile, small molecules, and those darker points are hydrophobic ones. So you can see they are heavily bound to the matrix because, of course, hydrophobic analytes free concentration is very low, while hydrophilic, as you can see, very poorly bound. So free concentrations are almost equal to the total concentration. And therefore, as you put the fiber in, you immediately equilibrate with the polar compounds and slowly extract the hydrophobic ones. So within half an hour, you collect both very effectively. And here, just to show you how does it work. So you compare the hydrophobic with hydrophilic. As you know, morphine is hydrophilic compound. So you can see there is no difference between extraction of PDMS and plasma. Plasma contains the protein and PDS not. So basically, you can see since I mentioned the 3 equal total, the amount of extracted in both cases is the same. Why in hydrophobic, you can see a huge difference. When there's no proteins, you extract more because in that case, 3 equal total. Why when you have a protein, some binding occurs and therefore actually fibers see much lower concentrations. So this is, as I mentioned, due to the binding. If you want to consider this process more fully, you also need to consider this dissociation rate constant and so on, which we have done as well if you look at our recent publications, which is actually shown here. Some very brief summary what this publication telling about is basically showing that because you have this balance between the competing extraction by the matrix compared to the partitioning onto the fiber, you actually extract the same amount or close to the same amount independent of the log P of the compound until, of course, the coating is not capable to extract anymore. That means if it's very polar, you can, compound is very polar, ionic, it will be not extracted by hydrophobic type of extraction phase, even a very polar one. So basically, because of this competing binding, you have this so-called balance coverage, and you can extract range of compounds very effectively. And we validated that on a number of experiments. Here, some of those initial validations, when you look at the bacteria growth, and we extracted them in a number of experiments, you can see we published in Journal of Metabolomics. What does it mean is basically this tool is very effective in extracting full range of polarity of the compound. So we can actually get metabolome profile. And the compound which extract from 100 to 1,000, now we can do even more depending on the porosity of this protection layer I mentioned about. As you can see, we detect from minus log, if you use a sorbent such as HLB, type of sorbent, we can detect it from log P minus 7 to all the way to 15. So we see all the compounds. However, because we extracted via free form, it's very clean extraction. So if you compare, for example, our approach, Spme, which is upper trace versus lower trace protein precipitation, you see you have this is blank after 40 rounds of protein precipitation, and upper is blank after 40 rounds of Spme. As you can see below, we have huge phospholipid band coming, while in upper chromatogram, which is basically multiplied background of the chromatin below, so it's basically 100 times more sensitive description. So it's basically you multiply the background by 100 times, and so you see you have very, very low background here. The Spme doesn't extract the huge amount of phospholipids as protein precipitation does, because we don't dissolve the phospholipids. You extract them via free form, but the solubility is so low, so we extract some of them to see them, but not enough to cause the trouble. Okay, so now from practical point of view, how do you use this technology? So the one of the approach for high throughput, we are using this device called Concept 96. So it's 96 blade system. So instead of coating fibers, we're actually coating blades to increase the surface area of the sorbent and therefore increasing mass transfer to the extraction phase. And having, as you can see, 4 different 96 wall plates. One of it, we perform the activation, the other one extraction, then rinsing, and then desorption, and then this 96 wall plate was analyzed, dissolved into it. It can go to the analytical instrument. Now, what's the advantages of this approach versus the SPE? So basically, the major one is because it's open bed system. Yeah, so it's basically no particles of the tissue can be trapped there. When SPE, when you percolate the sample through, it may occur. So that reasons why, as I showed you before, there is no matrix effect in this technology. You can do it also much easier automation, as shown here, and we can look at the larger amount of samples without worrying about breakthrough. So there are several advantages, and this is well described in the literature. Now, more recently, there was an improvement in this design, where you can see now there's a tower which holds 96 well plates, which are exchanged one by one. So first one was used for activation, then exchange it for extraction, then rinsing, then desorption. So such a device is a little bit less expensive. So it's available from a company called PS Technologies, if you are interested. Now, the other beautiful feature of Spme, as I mentioned at the beginning, is because you can sample directly from the source, is an excellent tool for in vivo Spme. And the primary reason for it is because you don't need to take the tissue out. So we call it chemical biopsy. We equilibrate the coating with the system and then pick up this. And if you make this coating in the form of coated fiber, so it could be very small, you show minimum damage and you don't take any tissue out. You just take chemistry out. So Barbara will explore this approach further, give you some applications. And again, the beauty about it is that when you remove this chemistry out, you also isolate this chemistry from the enzymes, and therefore there's no enzymatic degradation of the sample. So you combine it, the extraction was quenching automatically, and there's no matrix effect. So it's a very effective tool. So in summary, why it's in vivo Spme is very popular. First of all, it's metable coverage of the small molecules is very good. Eliminates suppression in mass spectrometry measurement. Captures unstable analytes, yes, because you equilibrate with real system and you can analyze immediately after the extraction. And then finally, because you are sampling the system with minimum damage. For example, if you do mice studies, you can use one mouse over the whole pharmacokinetic profile without doing much damage to the mice. Yes? So this improves the precision because inter-animal variability is eliminated. Here's the example. So here, one of our first studies, and for those who are interested, you can look into the paper which is listed below. When we compare standard approach on the right, which is the protein precipitation, so standard approach, and on the left you have two Spme approaches. One is equilibrium, one is pre-equilibrium, so short-term sampling, long-term sampling, 5 minutes versus 2 minutes, but you can see there is no difference in the profile, no difference even in absolute values. So there is no reason to do it direct, to do traditional methods where you actually need to draw a lot of blood from animals. And you can also do tissue sampling. So we recently published this paper in Chemistry, which drew a lot of attention, as you can see. And basically what it is, as you can see, there are two devices principally which we use in vivo Spme. One, lower one, you can see traditional one where you have a fiber in the sheath, in the needle, like I showed you before. And then the upper part where you actually have a coated needle, where the coating is in the recess. So, because now we can make those coating very robust, so the tip penetrates the tissue, and there's not much shear forces on the coating, so we can reuse this needle many times. So this is we think this would be the ideal medical device, and we are working with a number of surgeons and animal persons involved, researchers involved in animal studies. And finally, what I wanted to say, because the extraction is clean, we don't need to have chromatography. We can go directly to mass spectrometry. So this is really the ideal for moving everything on site, especially if you want to move it close to the surgery room or even doctor's office. So we can do it, have extractions, devices in the form of blades, tubings, meshes, and fibers, which we already discussed. And again, this has been discussed in this review paper I mentioned to you, and I will not talk about it in any more details, just indicate that this is an opportunity. Here I show you my group. Barbara will show you also her group, but I just wanted to show you what you see above is my website. Why I'll indicate to you, because we actually have the courses twice a year. The next one would be December 6th and 7th this year. If you're interested, you can sign up to actually experience the SPME, not only in lectures, but also in practice. We actually have a practical experiment there. And finally, there's also a conference called ExTech, and next year is going to be in Guangzhou, China. So again, you are very much welcome to attend it if you found this technology interesting, and if you're interested to explore other extraction technologies. So now I give my space to Barbara to continue the presentation.
So hello everybody. Now I will continue the talk by giving you some more information about clinical applications and some other bio applications like some cell line studies. So basically, whatever Janusz was talking about fits the clinical purpose. So as you can see here, SPME, as you already know, is used in headspace mode, also in direct immersion mode, and it both applies to clinical or bio applications. So basically, I will focus on direct immersion. Headspace is also used mainly, for example, for breath analysis, but also other analysis. Direct immersion is more new because we have right now biocompatible fibers. And thanks to that feature, it allows us to basically insert fiber directly into the tissue or blood. So the workflow that Janusz discussed with you also applies here. So as you can see, after extraction, the fiber is quickly rinsed, and then it goes to organic solvent or organic aqueous solvent mixture. And then we basically inject this extract to chromatograph coupled to mass spectrometer in most of the cases. So we are using two basic devices, two basic geometries of SPME. So fiber, which is the main geometry that we are using for tissue analysis because we want to provide minimum invasiveness to the system. And then also so-called blades. So this is basically thin film to improve the sensitivity. But also this is very common to be used in high throughput mode. And I will also show you some examples of such applications. So here I just want to show you what are the solutions of SPME to the demands that clinical applications have. So first, of course, we need to have very simple method of sample collection. Ideally, we don't want to collect any sample. If you want to, for example, do in vivo, as Janusz explained, it would be great. We don't want to deal with too much samples. So, of course, here we can either take very small amounts of the sample, very small volumes, because as you have already seen, the fiber can be really small, so the extraction phase can be really very small. So you don't really need to have a large volume of the sample. Or, as I mentioned, if you have in vivo type of application, you don't collect any sample, you just extract small molecules or other molecules if your extraction phase is designed for that from the system, and then you basically do not take out any sample. So the second demand is a simple analytical workflow. So if we really want to have SPME or any other technology in clinical lab, it cannot be very complex workflow. We want to have the least steps possible. And in fact, SPME is very simple. So of course it requires optimization. There is no magic here, but it integrates the sampling and sample preparation in one step. And if we also want to have metabolism quenching, for example, if we are looking for biomarkers, we need to quench metabolism immediately once we collect the sample. And here again, if we do in vivo, then we have also quenching integrated with 2 previous steps. So then it would be nice if we don't have to have for different applications, different methods. And I will show you a number of examples here where we can use SPME and pretty much with the same workflow. So we can use the same technology for many different applications. We can do target analysis, which is the most common for clinical analysis, but also we can do, for example, medical research based also towards biomarkers discovery. So we can do, for example, metabolome profiling or lipidomics analysis. And another demand from clinical point of view is application for complex matrices. Whatever matrix we deal with, so pretty much plasma, urine, serum, whole blood or tissue, these are really complex matrices. So as Janusz mentioned, and also I already mentioned in my part, we have already biocompatible materials which we use for production of the fibers. And basically we don't experience any matrix effect later on. We are eliminating large molecules from the system, and the matrix components, they do not attach to the fiber. Okay, so another 2 things which are necessary to be fulfilled in the lab is high throughput and automation. And in fact, as Janusz already mentioned in his part, we have the automated system that basically covers all steps in SPME workflow, which is preconditioning, extraction, wash, desorption, and also on top of that we can do evaporation and reconstitution. We also have an option to have a semi-automated system. So we are just replacing the 96 well plates, but we still have full control on the temperature. We have agitation of the system, and of course we have high throughput. And then we go and we have high sensitivity and wide dynamic range. And of course, it was already mentioned, we have a variety of sorbents and geometries we can play with. So by increasing, for example, volume of the sorbent, we have the increased sensitivity. So we have basically this high sensitivity. We are dealing with low concentrations. And of course, we can also play with the time of our extraction. So we can work at the equilibrium if we want to really reach the highest possible sensitivity, or we can work at pre-equilibrium. So then we actually have much shorter extraction time, but we sacrifice sensitivity. Then very important thing is to sterilize the fiber or the whatever device we are using, or to use them just a single time. And already from our experience, we know that we may autoclave the fibers. We can do basically we can do regular sterilization with autoclaving, but we can also use, for example, gas sterilization and of course alcohol sterilization, because in most of the cases the preconditioning is done in the mixture of methanol water. We may want to also have temporal and spatial resolution. So for that, we can, of course, use the small fibers, for example. So we can have number of sampling points just by inserting the fibers at different time points. And we still have very low invasiveness. The size of the fiber, the dimension of the fiber is approximately 10 times lower than the biopsy needle. So we can easily repeat sampling of the same animal or of the same organ number of times. Of course, in the same time, we can do number of extractions. So, for example, we can have the same time point of extraction, and we can have simultaneous sampling of different areas of the body. We can have fully quantitative method, and we don't have time today to play with calibration and explain you all type of calibrations we have, but Janusz already mentioned a few. So here we can really choose from a variety of different calibration approaches, and we can have fully quantitative data, and I will show you example in a minute. Of course, we want to have results very fast. This is the first thing that clinicians ask. So how fast I can get the data? Well, of course, if we are including LC run, it usually a little bit long, but as Janusz mentioned, we can couple a Spme directly with MS, and then we are actually hoping to have with this direct coupling to have instrumentation on site, so give the data immediately after sampling. And of course, it's very important. We don't want to have very large organic solvents on site. We don't want to deal with organic solvents if possible. And the Spme actually, because this is micro extraction technology, we basically have very minimum consumption of organic solvents. And if we have direct coupling with MS, it's actually even less. So now I will go through a few examples of clinical applications. The first one is focused on oncology. So right now we are working on phenotyping of brain tumors, and we are working towards intraoperative monitoring of biomarkers. So basically there are two goals. First is to propose a few compounds which differentiate brain tumors and their grades. So we are mainly focused on gliomas. And the other one is to propose new fast analytical approach to on-site diagnostic testing of brain tumors. So that's what I already mentioned, to basically have very fast answer in the operating room. Here you can see how we do sampling of tumors. So these tumors were just resected. This is still in OR room. So we are inserting several fibers to the tumor. On the left, you can see meningioma, and on the right, you can see glioma. So basically, we choose meningioma because this is, I would say, easy tumor to be sampled. They resected usually as the whole tumor, and this is very easily to be recognized by histological testing. So basically, we were using this tumor as a reference. So we were comparing our data with histology. On the right, as I said, you can see glioma. So this type of tumor has many subtypes. It's very malignant tumor. So this is the one that we are interested in to basically find biomarkers of different grades of gliomas and also subtypes of gliomas which are susceptible for given treatment or not. On the next slide, as you can see, we were optimizing the method for this brain tumors. So we were testing different desorption solvents. We have selected 7 millimeter coating for those fibers because this is the optimum size that basically fit our purpose. And then we were running the extracts using LC-MS, but high-resolution mass spec. You can see in the middle figures how many features we were able to actually extract. So you can see that even with just 7 millimeter fiber and half an hour sampling, we've been able to extract hundreds of compounds. And we always run in helic mode and reverse phase mode just to increase the coverage of analytes. So as you can see here, we are basically covering on each column from about 100 to a few hundred compounds. Then you can see that we were very successful in differentiating two different types of tumors, so basically gliomas from meningiomas. I don't have time to show you more data today, but we were also able to differentiate different grades of tumors and also when we compared with histological and genotyping data, our phenotyping were actually in very good agreement. On the right, you can see that we are able to actually pick up compounds of interest, so compounds which, of course, after running a larger cohort and after going through proper validation, we can actually propose some potential biomarker for different tumor types and different malignancy grade. So right now we are also focusing on lipidomics because we want to extend our studies to lipidomic data, and right now we are already looking into the data from more than 200 patients. Another oncological project that we are involved, it's actually in collaboration with Toronto General Hospital, is monitoring of doxorubicin. This is the drug which was used for high-dose local chemotherapy. So basically, that was animal study. So as you can see here, that was pig model. Our collaborators, the clinicians, they were actually looking into the concentration and distribution of this drug in the lung, so different lobes of the lung, because they are working on new method of drug delivery. So basically they were using the perfusion system to deliver this particular chemotherapeutic agents to the lung tissue where the tumor is located. So basically the lungs are isolated from the rest of the body, so the chemotherapeutic agent shouldn't actually go into different parts of the body. So the problem that the clinicians had was basically what is the concentration of the drug, because they cannot do that many biopsies over the period of this perfusion. So we were basically inserting the fiber to the lung, as you can see here. And the arrow points actually into 3 fibers located in one of the lobes of the lung. So this is on the left side of the picture. And on the right, you can see exactly the same spot after sampling. So as you can see, actually the method is not really invasive. There is no bleeding. There are just air bubbles coming up. But basically we were repeating the same procedure number of times. So you will see the results in a moment. Of course, again, this is the targeted method. It requires optimization. So we were looking into the effect of autoclaving. So basically autoclaving even improved the effectiveness of the extraction. We were also testing different preconditioning solvents. And what is very important, we, of course, here need to do calibration. So as I mentioned, we will not go into details, but here I just mentioned we were using external calibration. So as a surrogate matrix, we were using LAMP-LANG, and we homogenized this matrix. And basically, we were spiking at different concentrations of the doxorubicin, and we were extracting exactly using the same extraction time. And as you can see on the bottom right picture, we were comparing the results from the tissue collected in the hospital. So first we were extracting from this intact tissue, and then we homogenize the same tissue and we extract from this homogenate, and we were getting exactly the same data using the calibration curve prepared in the surrogate matrix. So then this method, of course, were fully validated. So you can see the result. We meet all the requirements. And then you can see basically the results during the perfusion, how the concentration and distribution of the drug was changing. So the clinicians were using 2 different doses of the drug. You can see first it was increasing, then at some point the drug concentration was dropping. So as you can see, we can get fully quantitative data using in vivo extraction, and for calibration using external calibration curve. Of course, in vivo solid phase microextraction was used for many other studies. Here you can see how we are doing extraction from the circulatory system, from the blood system. You can use either these needle assembled fibers, so you can insert them directly to the vein of the animal and then expose the fiber to the bloodstream for the extraction. Or you can also place this fiber through the catheter, which is standard for clinical application. You can also insert the non-assembled fiber through the Luer lock type of designs. So it's very flexible method. As you can see, you can just adjust it to your needs. So basically you can tailor your method to your needs and to the environment you need to work with. In terms of rodent studies, of course, such fiber, especially assembled fiber, is too big to be inserted directly to the vein of the animal. So in that case, you basically use one of the interfaces that was developed for that purpose. So here you can see that there are basically different designs. One which is in the middle. This is basically the interface, which is like a loop. So one end is inserted into one vein of the animal, and then the other end, it goes back to the animal. So the blood circulates through this interface. So you can basically insert the fiber to this interface, and you can also use, for example, syringe or other pump to provide some agitation by using a push-pull type of agitation. And then on the right, you can see that also this type of device can be one way. So again, you can use the push-pull with syringe, for example, to agitate the blood through the device. And then you can sample as it is shown on the picture. So again, some examples of the applications. So exactly these devices were used for extraction from the bloodstream of carbamazepine and its metabolite. So basically, the colleague of mine was using full pharmacokinetic profiling, full pharmacokinetic profile of carbamazepine and its metabolite. And it was the SPME data was compared with automated serial sampling and also terminal blood draw. So for those of you who are not familiar with the technologies, basically for this kind of studies, pharmacokinetic analysis, you really need to use number of animals. So you either sacrifice one animal per data point, that's for the terminal, and or you can basically take a volume of blood, but then the animal needs to recover for some time to be used for next sampling. So as you can see here, basically the number of animals needs to be quite large to get this full pharmacokinetic profile as shown on the picture. And if you look carefully into the data, SPME gives the same results, but RSD is actually a lot lower for SPME because, as Janusz already mentioned, we are actually eliminating inter-animal variability here. So first, we don't sacrifice that many animals, and the other one, we are improving the data by eliminating inter-animal or minimizing inter-animal variability. We are not limited to blood. On that picture, you can see that we are also using pharmacokinetic profiling of carbamazepine, but from the rat brain. And for this type of studies, we are also using microdialysis for comparison, because this is basically a gold standard, I would say, for brain studies. And as you can see, again, the data was in full agreement between the two methods. We are not limited to drugs, absolutely. We can also do endogenous compounds. So this is the example of the targeted metabolomics. So as you can see here, a colleague of mine, he was actually looking into different neurotransmitters during drug stimulation. Again, comparison of microdialysis showed very good agreement, but in fact, he was interested in 4 neurotransmitters, but because of low sensitivity of microdialysis, we only seen 2 neurotransmitters with microdialysis, but SPME allowed us to see all 4 of them. But despite the fact that we have seen 2, again, you can see that the data is in full agreement between the 2 methods. And while we already had the extract from that targeted analysis, we thought that it would be good to actually check how the untargeted metabolomics look like when we compare SPME and microdialysis. And as you can see from that table and the graph, there was a number of compounds that actually changed over the time after the drug stimulation. And of course, there was a huge difference between microdialysis and SPME in terms of coverage, because microdialysis is basically the method used for polar compounds. So non-polar compounds are not going to the dialysate, but for SPME, actually, we had much better coverage for non-polar compounds and slightly less for the polar compounds comparing to microdialysis. So basically, these 2 methods can be nicely used together, giving us a very broad coverage of analytes. And then I will go to the next project that we have done. Now, as you can see, we were using this high throughput and automated system. So basically, we were looking into the first pharmacokinetic of 2 drugs, tranexamic acid and rocuronium bromide, in the patients who received liver graft. And we had 2 groups. First was heart-beating donors and the other one living donors. So basically, especially for rocuronium, we were checking if this drug can be used as a marker of early graft dysfunction, because the dead donors, so basically heart-beating donor groups, are supposed to have worst metabolism of the drug, because the drug is mainly metabolized by liver. And in fact, we were right. So basically, this drug can be potentially used as a marker of early liver dysfunction. But we also looked into the global profile of the patient. So we were using exactly the same system for doing targeted and untargeted analysis, because for this type of analysis, you need to have really quite a large group. So we just, for this untargeted profiling, we just took a part of the patient's samples, and we just check if we have any type of separation. As you can see here, we were able to differentiate between the 2 groups, and also pick up some compounds which differentiated these 2 groups. So of course, after again running a larger cohort, if we confirm those compounds can be potentially used as biomarkers of early liver dysfunction. When we are talking about high throughput system, especially 96 plates, we can also think about different in vitro assays related to cell culturing. So here also we are using the system that I showed you previously, the fully automated system and also semi-automated system for doing cell line analysis. Well, usually we culture cell lines or doing any type of, for example, microsome assays in 96-well format. And this is also fully compatible with the device that we showed you previously. So here, for example, you can see that we can use this 96-well plates for culturing the cells and then doing time course analysis. So basically using the same samples for number of times, for extraction of number of times, and then we can get time course analysis, so we can observe what compounds are declining and what compounds are appearing in the system. So for example, here we were observing the effect of drug. First, we were extracting just from the cancer cells, and then we exposed them to the drug, and we were observing changes over the period of time exactly from the same wells. The standard methods, for the standard methods, you basically need to stop metabolism and you cannot reuse the same sample. You need to basically have number of samples for the experiments if you want to look over the period of time. Here we can decrease number of samples and reuse the same samples for different time point analysis. Also, this 96-well format, as I mentioned, is used for example, microsome analysis. So here we were looking into the metabolism of the drug combretastatin, which is a fairly new drug, and so far there is just a few metabolites known. So we've been using this time course analysis with SPME to figure out what new compounds are coming up when we basically incubate drug with the microsomes. We compared this data with electrochemical metabolism detection, and then we verified with in silico prediction. And as you can see, we've been able to increase the number of metabolites from 5 to 16 with the proposed approach. So basically, to sum up, you can see that SPME is very flexible technology. It can be used in high throughput mode, and it can be used in just the fiber mode to have very low invasive sampling. So you can easily sample very complex matrices, including tissues. You can get very nice and broad analyte coverage. The method is very minimum invasive if you use just this tiny fiber, which is basically the acupuncture needle size. Then you can have the time course analysis if you want to perform some extraction from either, for example, tissue without damaging the system, or, for example, you may minimize the number of samples and still have full idea about the changes over the period of time. Method is fully quantitative, so as I said, there is a number of calibration methods that you can use for that. And you can especially use them for different clinical but also medical research type of application. I prefer actually this method for tissue analysis over the traditional method, because we are actually having here true snapshot of the metabolism when we do in vivo studies versus homogenized studies. You can do cell culturing with your traditional 96-plate system, 96-well plate system, and you can basically reuse the same sample. And even at the end, you can perform traditional assays on the same samples. And of course, if we couple SPME directly with mass spec or other instrumentation, we can provide our clinicians with intraoperative rapid diagnostic tool in the near future. And I also want to acknowledge all my students, also Janusz Group, of course, our collaborators and our sponsors who are providing us with the instrumentation and the SPME devices. Thank you very much for your attention.
Thank you. Thank you, Dr. Bojko and Dr. Pawliszyn, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speakers will follow up with your questions via email. So let's get started. Our first question is, why did solid phase microextraction technology not find its way to general use in clinical laboratories yet?
Okay, so maybe I will take it. I think that there is not too many groups that they are using the technology. It's fairly new in bioapplications. But basically, we can see that for the last 4 or 5 years, the interest is really growing. So clinicians are really happy with the technology. So we basically work with many groups, especially surgeons, but not only. But I think that, well, right now there is only one company which provides the biocompatible fibers. So that may be one of the problems. But I think that also people are they do not treat sample preparation as a science. So they are very focused, especially in medical area, they're very much focused on the traditional methods. So I think that this is one of the problems that basically the medical area is very difficult to go through with some new things, and SPME is definitely one of such things. But I can see from our experience that actually more and more people is interested. So I hope that in the nearest future it will be much more people, many people using that, much more than today.
Thank you for that. Next one is, what is the future of the SPME technology?
Yes, basically, SPME is a very simple way of performing sample preparation. The future is bright. The issue is it's a new kid on the block. So, in fact, it's kind of related to the previous question. Anything which new looks very difficult because the device looks different, the procedure is a little different. Calibrations are not that different, but they're somewhat different in some cases. So there's a lot of reservation. However, the format is so simple that it can actually potentially solve a lot of problems. For example, just to give you the idea, one of the future I see for the technology, and I mentioned that this picture was a drone, is that actually we can connect patients in environmental pollution, even in remote places, into the central labs. So basically, the devices are so light because they actually enrich the sample in a small volume, like small fiber or some blades. We also use the meshes or the format. You can then easily transport it to central lab. And therefore, remote communities, for example, northern Canada, Africa, the issues of providing the services to those communities could be very easy reached by those technologies. So this is definitely one of the future. In addition, of course, the whole lecture is the future because not many labs are using it.
Thank you for that. Next one is saturation of the coating a problem?
Yes, I mentioned that in my part of my presentation that because we have this process of extraction via free form, and free form of analytes is controlled by the solubility of analytes, and the compounds which are hydrophobic, which actually has a poor solubility in aqueous matrix, which is most of the sample constitute of, basically limits this possibility of saturation. When I talk to a lot of scientists, because of the paradigm, that's what I'm saying, you need to think outside the box. This was the first picture I showed, because they always think such a small device, there's so many other compounds, they can do so much damage to them, but in fact they don't. And in fact, if they did, we'll die, because a lot of systems work in a very similar way. The proteins which carry the analytes are not saturated by other analytes because the equilibria prevent them to do that. So basically the same fiber equilibrates with the system, but because the equilibration occurs via free form in aqueous media, the saturation never occurs because the compounds which can saturate hydrophobic analytes are not very soluble. So the process of extraction is extremely slow. So within a short period of time, which we perform the extraction, there's not enough analytes reaching the fiber to saturate it.
Thank you. Looks like we have time for just one more question. How is solid phase microextraction calibrated?
Yeah, so this is, as I mentioned, one of the standard blocks because some people say, well, how do you calibrate the process, which is completely different than exhaustive extraction? In exhaustive extraction, you calibrate on volume. So you take 1 liter of water or 1 milliliter of blood, and then whatever you extracted from this 1 liter or 1 milliliter, and you divide by the volume, gives you the concentration. Here, it's a little bit different. So basically, you need to develop a calibration profile called matrix match calibration, which is basically the same things what we are doing even in exhaustive method. Because the issue is if you have a complex matrix, you still validate that you have exhaustive extraction by using the calibrants that adulterate the analog, of the analytes and so on. It's the same thing. It's the same thing here. You can use the deuterated analog to calibrate, to basically, in a sense, find the K value or some other parameter, and based on that, you can calibrate your analytes. Because if you know the K value for a given matrix, you can do that. There's also a very interesting way of calibrating called calibrant in extraction phase, because remember, it's non-exhaustive technique. So when you place the fiber in the coating, in the sample, actually the analytes can also leave, or the calibrant can leave the coating to go to the matrix, because it's a multi-phase equilibrium. So in that case, you actually can calibrate for the convection conditions. That's because when you have some extraction phase in contact with the matrix, the amount of ions extracted depending on the convection conditions and the concentration. So you can ratio out those convection conditions. As I said, so in a sense, they are very similar calibration approaches at the traditional method.